You may have heard the term sickle multiple times during the span of your life, whether it was during a biology lesson of 9th grade or the genetics unit during college biology. You guessed it. In this video, you'll hear it once again and learn about the origin of the sickle allele, including where and when it emerged. Let's dive into it. The sickle cell trait describes a condition in which the person has one abnormal allele of the hemoglobin beta gene, but does not display the severe symptoms of sickle cell disease. Why? Because this trait has recessive lethality, meaning if the person carries 2 copies of the recessive allele, it is very dangerous and fatal. The sickle allele is one of the variants in the hemoglobin gene that protects individuals against malaria, a disease caused by the bite of infected mosquitoes. This sickle allele is under balancing selection because of its heterozygote advantage. Balancing selection is when selective pressures select against the 2 extreme phenotypes and select for the intermediate or heterozygous phenotype due to its advantages. The sickle allele is selected for because it protects against malaria caused by recessive lethality. And Plasmodium falciparum, a parasite that is one of the species of Plasmodium that causes malaria in humans and can be transmitted by the female Anopheles mosquito.
But where did this allele originate from, and is its emergence recent? According to other various studies, the emergence of the sickle allele was either recent, meaning it occurred within the last few thousand years, or it is quite ancient, anywhere from tens to hundreds of thousands of years old. Additionally, some studies suggest that each haplotype, or a group of genes within an organism that was inherited together from a single parent, represents an independent occurrence of the same exact mutation in its corresponding geographic region, and other studies suggest that the mutation occurred only once and was passed on. With these pieces of information, possible regions of origin for this allele are Equatorial Africa and the Middle East. In this research study, the origins of the sickle allele was found using whole genome sequencing, the process of determining the complete DNA sequence of an organism's genome at a single time, from 2,932 individuals from a number of genome projects in Qatar. Through this sequencing, 156 sickle carriers were identified. The haplotypes were then determined by using phase sequence data to find a single origin of the sickle allele approximately 7,300 years ago during the Holocene wet phase, or Green Sahara. For context, the Holocene wet phase was an extended period of wet and rainy conditions in the climate history of northern Africa lasting nearly 4,000 years. It was both preceded and followed by much drier periods of time, suggesting that mutations could have arisen to help already existing species better adapt to the rapid and extended change in climate. By clarifying the origin of the sickle allele, the process of identifying the allele can be improved and simplified. Improvements to the process of finding this particular allele are important because the gene mutation affects between 1 and 3 million Americans and 8 to 10% of African Americans. Scientists Daniel Schreiner and Charles Rotimi, who wrote the paper on the origin of the sickle allele, both work for the Center for Research on Genomics and Global Health.
The authors analyzed their data using 3 sets of markers with increasing precision. First, 10 unique classical haplotypes for the sickle allele were identified. Take a look at this table. The leftmost column shows acronyms of regional sample data, and the column headers show names of classical sickle haplotypes. As we can see, for example, the Benin haplotype was a predominant haplotype in samples of Isan and Yoruba from Nigeria, and so on. Using more precise markers, 5 unique haplotypes were identified. One haplotype, the ancestral allele, was found at all markers. Fine-tuning the markers even more, 59 unique haplotypes were identified, the most common of which carried the ancestral allele. The ancestral allele accounted for, in fact, 68.5% of all haplotypes in continental Africans. It was the most frequent haplotype in all 26 samples of the 1,000 Genomes Project and was the only haplotype in 15 of those samples. Modeling the balancing selection of that allele indicated that an equilibrium frequency in the population was 12.0%. The researchers asked themselves how many generations would it have taken for this equilibrium frequency to be reached. Factoring the recombination mutation rates, the age of the sickle mutation was marked as 259 generations, or approximately 7,300 years.
The results concluded that the common ancestor of the various haplotypes with the sickle allele originated about 9,200 to 12,000 years ago, in or around present-day Nigeria. They then found when the actual sickle mutation formed. This was found by investigating historic records, simulations of a balancing selection, patrilineal date, and ancestral combination graphs. The data in these records indicated when each haplotype separated from the others. It took about 2,400 years for the balancing selection to increase the frequency of the sickle allele to about 12%. These findings are consistent with the assumptions that no gene flow occurred between the different haplotypes due to the geographical isolation as the different haplotypes formed in different regions.